Okay, it's just about 3 o'clock now. I'm gonna get started with some quick reminders. So welcome back to our third and final session of the fourth annual Delaware Watershed Research Conference. The first thing, just a reminder again, please keep your microphones on mute unless you're speaking, and if you're speaking, you can take it off mute on your own whenever you're ready. Two, if you have any questions, please put them in the chat. If you can, put the speaker name and your email address just in case we don't get to them. We've been getting to pretty much all the questions, I think, but that way I can give that to the speaker if we don't get to it and they can contact you. The third thing, our moderator for this session is Catherine Christopher from the Academy of Natural Sciences. She will be giving the speakers warnings and may politely interrupt if we go over. And so with that, I'm going to hand it over to Catherine to introduce the first speaker.
Thank you, Catherine. Welcome everyone to the third and final session. As my fellow Catherine said, I'll be holding up little sticky notes. Hopefully the presenters could see this, just to give a heads up when time is running low. I have stickers for 5, 3, and 1 minute left. And yes, if we have some time to go over a question or two at the end of each talk, we can, but otherwise we can just address them in the chat. And if you feel comfortable responding to a question in the chat, go right ahead. Otherwise, we'll get them to you. To start off our session this afternoon, we have the Academy's own Stephanie Kroll, who is going to give a talk entitled Setting Targets for Watershed Health in the DRWI and Related Research on the Projected Effects of On-the-Ground Actions. Steph, take it away.
Thanks, Catherine. And thanks, Catherine. So this is the work that we're doing as a team and in close collaboration with especially John Jackson and some other folks at Stroud Water Research Center. And it's funded through the William Penn Foundation for the DRWI. So I want to just put up front those thank yous. Next slide, please. The biggest challenge we are facing in the questions that I'm going to discuss are what are these ecosystems going to look like when they have improved from all this great action that our partners in the DRWI are doing? And so we want to think about that image of a river, what it should look like, what a healthy river should be, because that idea is going to guide some of the actions that people are doing on the ground. We really want to base it on existing data and then have it informed by research. Next slide. So when we think about how to measure this, we think about all the interactions here and the sort of bottom up relationship, which is actually in the top of this diagram. So land use or land cover is influencing, all of these parts of habitat and chemistry, and then those are influencing biotic integrity. Which I detail more in the next slide. Oh, don't try to read this. Catherine, can you just hit the button? I forgot this animation was here. This is just when we have been thinking about all the different ways that the farmland is affecting aquatic habitat and chemistry, and then what those effects might be on the biota. So this is just a brainstorm for us to think about, and I'll address some of these metrics and effects throughout the presentation. Next slide. I'm obviously not used to doing this. So hey, my colleague Haley Oakland and I, with the help of a lot of interns, wanted to see what the literature said about the effects of best management practices and how streams might be responding. And so what we found was that it's not as well studied and there's not as much of a consensus as we might hope. Can you click, Catherine? And so this table just shows for different categories of agricultural best management practices, what the ranges in reductions in nutrient and sediment were. And so that's the number of studies and the mean underneath. But you can see for livestock BMPs, a huge variety of results from 8% reduction to 85% reduction in nitrogen, similar for phosphorus and sediment. So the crop land ones we categorized, which are cover crops or nutrient management, had narrower ranges, but still pretty wide ranges. So it is quite a challenge for us, even with the models that Lynn is working on with Barry Evans and other folks, to those coefficients going in to predict efficiency and effectiveness of these practices is part of the challenge. And then the other challenge is really just that, you know, everything varies so much across different geographies that there aren't great studies really just giving us examples in the Delaware Basin that say, yeah, we think that if they're using cover crops, there could be this much reduction. And so we're working with these levels of uncertainty and just trying to do our best to say, what will the system look like? Next slide, please. So again, don't read this. This is an exercise we went through a few years ago, just thinking about our different indicator sets in the different clusters and what the metrics that we're seeing look like now for diatoms, macroinvertebrates, and fish, and what we would like to see as their improvement in the future. So, you know, you can just take a little glance in the agricultural clusters, which are these top 4. You know, we'd like to see reduced dominance by tolerant taxa, by nutrient tolerant taxa. For diatoms, for macroinvertebrates, we'd like to see improvement in all metrics. And for fish, we have some objectives of greater diversity and more stable functioning, and maybe some reproducing trout if we're lucky. And then in protection, we would just like to see maybe a little improvement, but mostly maintenance of those conditions. I'm happy to share this stuff. It's in a report we wrote in 2015. Next slide, please. So just to look at what one of the variables we think about is changing communities over time. And so Marie has shown, you know, that variability over time and how we can use that to decide, to observe whether something has come out of that variability. And the same goes for our biotic communities. So this is work by Dave Keller and Alison Stocklosa with Rich Horowitz, where we have to think about, you know, fish are moving, we've got sampling error, and then hopefully we'll see changes due to restoration. And of course, there's a lot of other things that can be changing the communities. So first of all, just understanding that variability so we know when a community has really changed from just that interannual variability. Next slide, please. And so Dave's team looked at 2 different similarity index. The Bray-Curtis is basically species turnover, or the changes in species between the different years that they sampled. So just looking at their top 5 fish, which were pictured here in the study, these were all native and they preferred like running water benthic habitat, so in between the rocks. Some of them can be difficult to estimate because they outsmart our electrofishing equipment and net gauges. And some are just naturally, you know, variable. So the patterns are hard to tell anyway. But Dave's group thought in order to consider variability in projecting change, we want to just accept that it's variable and maybe just focus on some of the fishes that have more stable populations. Next slide, please. The Jacquard Similarity Index showed a different result in that it looks at the changes in species proportions. And so for these, the top, out of the top 5, 3 of them are non-native, and the warm water or prefer slow moving waters. And so the brown trout signal also is probably affected by stocking. So here again, Dave's group decided, let's focus our attention on those components that are more stable and just keep track of when we have a lake nearby. So I'm going to switch over to macroinvertebrates and diatoms for the next slide. Sorry, not yet. And so this just means when we're thinking about detecting change, we really have to consider the preservation sites differently from restoration sites and really look at that variance. Okay, now next slide. And so when we think about why are we measuring macroinvertebrates, fish, and diatoms, these different indicators could tell different pieces of the story on watershed health and the resilience to change. And then when we think about the data we are using, so metrics are these variables that are calculated from the count data, which is how much we have of each thing. And metrics describe structure, so how many different creatures we have, and then some ecological function. Metrics can be a simplification of the data, which is useful for communication, but it does mean a loss of data. And so a study that Haley Oakland and Allison Minerovic and I have just submitted for publication, next slide, is looking at, can we use these metrics that are so loved in indices of biotic integrity and for other reasons, or do we really need to go to which different species or genera are where and what does that mean? So we compared using these two data sets, each in relationship to environmental variables, and ran a Procrustes analysis to compare the two ordinations. And so the next slide shows plotting the differences in the health assessments of sites based on these two data sets, the counts and the metrics. So this x-axis represents our gradient of conditions. On the negative end is actually the highly forested sites, and on the positive end are high percent agriculture sites. And then the y-axis shows how different the sites are based on the metric classification or the counts. And so this U shape tells us that those two approaches to analyzing the communities are most similar in the in-between sites, in the intermediate agriculture sites, but they're very different in the highly forested sites and also the very highly agricultural sites. Next slide. For diatoms, we found a slightly different pattern that in the forested sites, there was also a big difference between the two types of data in classifying our sites, but that for the rest of the sites that were experiencing more impairment, they were much more similar. And so these studies just are sort of a cautionary tale that people really love metrics, they love using them, but we can't necessarily say that those are going to be the best ways to show what the condition is right now, and therefore not the best sites to set projections for improvement. Next slide, please. But we still can use them to set some goals. So these are the changes that we would like to see in some key metrics if restoration shows success. And they're a little bit conservative, so hopefully we'll just blow them out of the ballpark, but not sure about that. Next slide. I'm talking fast. I'm trying to give you guys a really nice flavor of all the work we're doing. So then when we look back at communities and species level analyses, or genera as it is for macroinvertebrates, we're using now in a paper we're working on a threshold analysis where it basically says which specific taxa respond. So this x-axis is that same environmental gradient. And so those on the negative side are present in a forested area. And then as those black dots decline, that means that's where along that gradient that these taxa might disappear because they just can't exist in those worse conditions. And then the white circles are showing the taxa that are sort of benefiting from a loss of forest and an increase in agricultural cover. So again, don't read this. I'm happy to share anything. And we did this for macroinvertebrates. And then on the next slide, we also just pulled out some of these just to give you like a representation of if we had these taxa in urban and agricultural land, like what might the assemblages look like over time? So you can think about it for individual taxa or the community. Next slide. And then we did the same thing for diatoms. The interesting thing with diatoms is they really do directly respond to nutrients and sediment. And so for these and for the macroinvertebrates, we look at the whole gradient and then we pull out single indicators and run the same threshold analysis, which we're working on right now. Next slide. So that's some of the work that's ongoing, certainly not all of it. A project that is just getting started right now that I'm really excited about, and Mike Slattery referenced that some of us have been awarded grants through the Delaware Watershed Conservation Fund. So we're looking at headwater biodiversity and resilience to climate change in the upper Delaware Basin. We'll be collecting macroinvertebrate communities and looking at these specimens to ID them, but also doing DNA metabarcoding to look at cryptic diversity. And then in some places, we'll be collecting eDNA to see if we can identify any vulnerable taxa, any of those endangered mussels, maybe some eastern brook trout that we want to protect. And we'll also be assessing in-stream metabolism. So this is a really exciting project. We're really just starting like November 1st with some great collaborators throughout the basin. And I'm also happy to share more information on that. Next slide. All right. So that is the end of my presentation, but I did want to say Marie Kurtz and I are bringing together a scientific advisory committee to advise on, the direction of DRWI science and facilitate collaboration. We have a lot of data. We have a lot of different partners who are from, research groups, and we have a lot of practitioners who are really interested in using the research and in informing what we study. And so because of this, we are starting this scientific advisory committee. If you would like to join, we plan to meet about every 2 months or quarterly, just talk about the different work that's going on, maybe find some great collaboration. And Catherine Longwell, who was on here, will be bringing this together. So you can email her directly at that email address, which we'll also put in the chat. And you can ask about that from her or I. Next slide, please. And I'll throw my email in the chat. I think most of you know where to reach me, but this is our Patrick Center team. And a lot of these, everybody works with some of their time on the DRWI. So just wanted to get all the faces up there and say this is really collaborative within the Academy and outside the Academy. And we just want to make the most of the work and all the great relationships we have. So thanks for listening.
Excellent. Thank you so much, Steph. I will let Catherine set up the next talk, but in the meantime, we do have a question in the chat. What were the predicted numerical increase in impairment score derived from? Was it what you would like to see, or was it based on some empirical evidence? I think we have a few minutes for you if you want to tackle that.
Okay, I think you might mean the metric improvement that we're hoping to see if the restoration is successful. Yeah, thanks, John. That is based on our data. It's based on the ordinations with the metrics, and then just breaking them out in sort of, I can't remember now, it's quantiles, and looking at what's the average value of number of mayfly families in the best and the intermediate and in the worst sites. And so what is that difference now, and how much of that difference does it seem possible to sort of span with improvement? So it is based on our data, but I will say they are conservative. So I might have seen higher numbers in some parts of the data and just brought it down a little bit so that it seemed reasonable.
Great, thanks.
Oh, and thanks.
Yeah, there is another question there if you wanted to answer that via the chat. I think
Sure.
we're going to get started on our next talk. Next up, we have Lesmes Alejandro Mora Jerez from Villanova University. And his talk is entitled Lessons Learned from Monitoring 4 Headwater Streams in Delaware County. So whenever you're ready, Lesmes, you can take it away.
All right, good afternoon. So I'm Lesmes Alejandro, and today I'm presenting lessons learned from monitoring 5 headwater streams in Delaware County. This presentation is divided by 3 sections. Section number 1 is talking about the introduction, context, and motivation, as well as the research approach. For section number 2, it's about the area of interest, as well as monitoring sites and studied streams. And for section number 3 is methodology, preliminary results, and research plans and future work. So for section number 1, so as you can observe through this image, this is a natural watershed. However, the redevelopment of the increase of impervious surface area through urban settings, it can also create an increase of urban runoff that ultimately discharge into the stream, and it can change the hydrology and the hydraulics of the stream. For example, it can carry, or it can move some pollutants and contaminants from the traffic and it can incorporate those into the stream and change the water quality. It can also move the salt applied during the winter, and it can change the electrical conductivity and chloride concentrations into the stream. Also, it can produce change of the temperature at the stream. So the increase of urban stormwater runoff can not only impact from water quality perspective, but it also impact in terms of the amount and velocity of water that is going to move through the stream, and therefore it can impact in the amount of sediment that is going to move from upstream through downstream, and ultimately is going to impact the biological communities. So based on this situation, we need to understand if the stream is capable of sustaining itself. And if we understand this, we can define if the stream is really healthy or not. And one way for studying stream health is actually analyzing each level of the stream function pyramid framework, specifically by studying the biology, the physicochemical, geomorphology, hydraulic, and hydrology. And we need to understand how different rainfall scenarios can impact at each level of the stream function pyramid as a result of the different peak flows. One of the motivations of this study is that each watershed is different. In fact, we can find developed watershed signatures. Some of them are completely pervious. Some are 50% pervious, 50% impervious, and others are just completely impervious. So based on each developed watershed signature, we want to see how each level of the stream function pyramid is impacted, and how this can change the stream health as a whole. So in order to understand, in order to study the stream health, we are analyzing each level of the stream function pyramid. By answering the question, what are the lessons from studying each level of the stream function pyramid framework in 4 streams in Delaware County, Pennsylvania? Therefore, we are analyzing the data specifically at the hydraulic and hydrology level. We have a flow meter for each of our monitoring stations, and it collects the flow rate and depth. Also for the geomorphology, we collect a sample from the stream bed, and we analyze the grains of distribution by performing sieve analysis. In terms of the physical-chemical, for example, we have a fantastic group in Villanova University who performs all the tasks for determining the water quality parameters at the Villanova Water Resources Lab. And we collect a storm flow and base flow samples, and we determine all the water quality parameters. Also, we find the dissolved oxygen concentration and conductivity, electrical conductivity and temperature from dissolved oxygen and electrical conductivity probes that we have for each of our monitoring sites. And finally, for the biology, we work with Dr. Kemp from the University of Baltimore in order to perform electrophysics as well as microinvertebrate samples. So as you can observe, we have been analyzing each level of the stream function pyramid. Now, what are they located? What are our monitoring sites? So the main area of interest in this case is the Granite Run Mall. The redevelopment of the Granite Run Mall provided a fantastic opportunity in order to test the efficiency of old and new stormwater control measures that are located within the Granite Run Mall. The urban runoff usually is discharged in the Crum Run, which is the impacted stream. This is the discharge at Crum Run upstream. And here you can observe the port of pavement, as well as enlarged detection basins, which are basically the stormwater runoff, the stormwater control measures that we have at the eh, at the Granite Room Mall. Now, we have 5 in-stream monitoring stations, which they are located in Media, Pennsylvania, specifically one downstream of the Crum Run and another upstream. We have also in the Small Run, the control site, and also 2 reference sites located in Dix Run as well as Rocky Run. We have meteorological station, as you can observe through this picture, and this is located at the roof of the Bancrest High School. And we have 5 in-stream monitoring stations, which basically collect the flow rate, the depth, temperature, conductivity data from our sensors. It collects the data, everything into this data logger, and transmit the data to our database. This table shows a summary about each stream that we are studying. And basically we have the Crum Run, which is the impacted stream. It's considered as urban watershed, and it has the high impervious surface area with 43.2% from upstream and 26.7% from downstream. On the other hand, this Small Run is the reference stream, and it has the lowest impervious surface area with 3.3%. And we have Dix Run and Rocky Run with a 14.3 and 7.3% of impervious surface area respectively. For section number 3, so I'm explaining about methodology, results, and lessons learned from this research. So as you can observe, we have been analyzing each level of the stream function pyramid. So by evaluating the hydraulic and hydrology, we studied the stream freshness index, which is a ranking that allows to identify the hydrological response of the stream during different rainfall events. So we use an equation developed by Baker et al. 2017. And this equation allowed to determine the Richard-Baker stream freshness index, which is that hydrological response. And it's basically based on the difference of the flow rate for each time step divided by the total flow rate during this precipitation event. When that we applied this equation, we found some interesting results and lessons that we learned from those results. This graph here, this hydrograph, shows that impervious surface watershed tends to have, they tend to show higher peak flows as well as a shorter lag time in comparison to those watersheds that are pervious. In regards to the results, so those results that we found suggest that high impervious surface area, high impervious surfaces areas in watershed imply an increase of stream freshness index. Therefore, a high impervious surface areas could produce an high hydrological response during a rainfall event. On the other hand, this Small Run, which has the lowest impervious surface area with 3.3%, also has the lowest stream freshness index. One of the lessons that we learned is that we need to create a new equation in which we consider other driver factors that could impact on that hydrological response, such as the slope of the watershed, for example. And another lesson is that there is a challenge to collect the flow rate data in natural streams, because during a higher precipitation events, the flow meter can be displaced. So we need to do a specific setup for making sure that actually the flow meter can record the data continuously. Now, going directly to the geomorphology level, here you can observe, for example, the watershed at the Crom Run. And we determine the average annual soil loss in Crom Run. And this shows on this map, based specifically those brown dots, that we have a specific amount of erosion and sediment that is going into the stream. And this analysis by using GIS and the Russell equation, it makes sense because we don't find any erosion for the areas that are impervious, such as the gray areas located in this watershed. So in addition to these value or this data, we also use the bed slope, channel width, grain size distribution, intermittency, and theoretical flow discharge. And we use all of them as input data into an aggregation degradation model in order to know how much erosion could occur in, in a specific watershed. And one of the lessons from this analysis is that we need to calibrate the model and we need to readjust the intermittency value based on the collected data. Here is a result that shows, for example, that the Crom Run upstream can lose specifically 0.2 meters of the stream bed over the next 50 years based on this model, and the system tends to record to the equilibrium 180 meters downstream. Regarding with the physical chemical, on the other hand, you can observe here some results. So for example, for electrical conductivity, we found that those watersheds with high impervious surface area, such as Crom Run upstream and Crom Run downstream, they also have the higher electrical conductivity, while the Small Run, which has the lowest impervious surface area, in comparison to the other watershed, it also has the lowest electrical conductivity. In regards with temperature, during June 2018, we can observe the peaks of temperature from Crom Run upstream, which has the highest impervious surface area, while this Small Run, which has the lowest impervious surface area, also has the lowest temperature values based on these results. And finally, for total phosphorus concentration for both base flow and store flow events, we found that Crom Run upstream, which has the highest impervious surface area, also has a high total phosphorus concentration for both base flow and storm flow events, while the lowest total phosphorus concentration is found for the Small Run, which is the watershed with the lowest impervious surface area. One of the lessons that we found from this is that we need to normalize each parameter based on impervious surface area. We need to compare each parameter from each stream based on a time series. Also determine a baseline for each parameter and compare a stream by stream, and evaluate how rainfall amount, time, duration, intensity, and frequency could impact the maximum concentration differences among urban stream for each water quality parameter. And finally, at the biology level, we determine the fish species richness, and here you can observe in more detail the graph. Something that we found is that the Crom Run upstream, which has the high impervious surface area, it also has the poor level of fish species richness. Now, for Rocky Run, which is not the lowest impervious surface area, and it has the higher fish species richness. One of the reasons of this is that actually the Rocky Run has a drainage area that is larger in comparison to the other watershed. Rocky Run, sorry, this Small Run is the watershed with the lowest impervious surface area, and also has the highest fish species richness. And based on the ranking, this is considered such as a good a good fish species richness in terms of this parameter. And in the case of Dix Run, in the case of Chrome Run downstream and Dix Run, those watershed they have a fish species richness that they can be considered moderate. So those considerations about to define when the fish species richness is poor or fair or good is calculated based on the area of the watershed. And also I can provide that information in the chat for additional clarification. And finally, one of the lessons from the biology level is that geomorphology changes could be a driver factor that alter fish species richness. There could be a significant relationship between fish species richness and amount of impervious surface area, and drainage area plays an important role for fish species richness level. And I would like also to add that it's very important to match this data, the electric conductivity, temperature, the soft oxygen concentration, with this fish species richness in order to see if there is a correspondence between the water quality data and the biological data. Finally, I would like to acknowledge Villanova University College of Engineering, Sea Grant Foundation, as well as William Penn Foundation for providing all the help and support for making this research possible. Also, I want to say thank you to all the undergrad, grad, faculty, and staff, undergrad and grad students from Villanova University, and all the people from each of the sites in which we set up our equipment. I want to say thank you to all of them for allowing us to do research in those specific sites. Thank you very much, and I will be more than happy to answer any questions that you have.
Thank you so much, Lesmus. That was really wonderful.
So my name is Shane McLaughlin, and I worked with Stratton McGinnis and our principal investigator, Heather Murphy, to work on understanding the health risks due to recreation along the Wissahickon Creek, the preliminary data from summer 2020 sampling. We're part of the Water Health and Applied Microbiology Lab, or the WAM Lab from Temple University in the Department of Epidemiology and Biostatistics. Next slide. So a little bit about our team. So we're led by Dr. Heather Murphy, who's here today to potentially answer any questions. Our research areas include research in groundwater quality, water sanitation and hygiene, stormwater management and surface water quality, as well as drinking water distribution. Next slide. All right, so a little bit of background. So recreations such as swimming, wading, and fishing are frequently observed along the Wissahickon Creek. And previous research has found that recreation along some water bodies may present a health risk. So here's a picture of people swimming in Devil's Pool. Okay, next slide. And a little bit more background. So recreation along water is impacted by combined sewer overflows, also called CSOs. In Philadelphia can represent a health risk, but research is lacking around the potential health risks due to recreation along the Wissahickon, which is not impacted by CSOs. So our research was meant to figure out the health risks that might be associated with those that are not impacted by CSOs. Next slide. So our two basic research questions were, what is the risk of illness due to recreational activities observed at sites along the Wissahickon Creek? And also, how does this compare to the risk calculated due to recreation along CSO impacted sites in Philadelphia? So in this presentation, we're going to first present the preliminary water quality data collected at sites along the Wissahickon Creek. Second, we're going to discuss the previous research that has measured the health risks due to recreation along CSO impacted sites. And three, outline the next steps for this project. So Here's a slide that shows where we sampled this summer. So the Wissahickon sites are in the northwest corner of the map. So there's 5 locations that we sampled at: McGarge Dam, Valley Green Inn, Rope Swing, Devil's Pool, and Kitchens Lane. And on the next slide, I have some photos of those. And then we also sampled at 2 other sites at Cobbs Creek and Tacony Creek, which are also on the map. And those are impacted by CSOs. But for this presentation, we're going to focus on the Wissahickon sites only. So here's some photos of the places that we sample at. So the first one's the Valley Green Inn. We sampled right beneath that bridge, the McGarge Dam, where fishing has been observed. The Rope Swing, which you can see is a popular recreation spot during the summertime, and Devil's Pool, which is also another popular recreation spot. And then we also have Kitchens Lane, which is known for its dog beach. And then we have the 2 other sites that are the CSO sites, which is Cobbs Creek and Tacony Creek. So what are our methods? So we did a total of 66 samples at the Wissahickon this summer, collected between May 2020 and September. We collected wet and dry samples, and I'll explain more about why we did that later. But a wet sample meant that rain had occurred within 24 hours of the sampling, and dry meant it was at least 24 hours since the last rain event. So we collected 2 forms of samples. Collected 1 liter grab samples so that we could analyze them using culture methods in our lab for E. coli, total coliforms, enterococci, and fecal coliforms. And we also did a 50 liter ultrafiltration to measure for enteric pathogens. So traditional fecal indicator organisms, FIOs, are used to set regulations. These are total coliforms, E. coli, enterococci, and fecal coliforms, like I said, and they're detected via cell culture, so we were able to perform those in our own lab. But they don't always tend to correlate with human pathogens. So they can suggest a health risk, but they cannot be directly linked to a health risk. So for our grab samples, we perform membrane filtration, which is passing through the sample through a filter under a vacuum. The filter is then removed and incubated on an appropriate agar for 24 hours. And then we count the colonies that grew on the agar. And we do that at different concentrations. And then second of all, the ultrafiltration method. So we use these dialysis units, Rexseed dialysis units, which have membranes, filled with membranes in the water that passes through it. It has very small pore sizes, so it captures viruses, protozoa, helminths, and bacteria. So 50 liters is passed through that using a peristaltic pump from the surface water. And then after 50 liters runs through, we close it up and then ship it off to a lab, a USGS lab, for analysis. So like I just said, the ultrafilters are sent to the USDA lab for qPCR or RT-qPCR, and they're analyzed for the following pathogens: adenovirus, enterovirus, norovirus, Campylobacter, enterohemorrhagic E. coli, Salmonella, Shigella, Giardia, and Cryptosporidium. And then we also analyze, well, the lab also analyzes for genetic markers for human sewage, which is more specific than the fecal indicators that we measure within our own lab. These include HF183, which is a gene marker for Bacteroides, which inhabits the human gut. Pepper mild mottle virus, which infects pepper plants, but since humans eat peppers, it can be an indicator of human contamination. The human polyomavirus, and SARS-CoV-2. All right, so here are preliminary results. They're only preliminary because we haven't received the sample analysis back yet from the USGS lab. So these are just our culture method results. So as you can see for the first side of the slide is the dry sample. So no rain had occurred within 24 hours. And the sites are Devil's Pool, Rope Swing, Valley Green, McGarge Dam, and Kitchens Lane. So you can see also at the bottom that there's the guidelines for the concentrations for safe recreation. So you can see that for E. coli and enterococci, which are more human-specific indicators, for Devil's Pool, there was not a problem on average for the median. But for Rope Swing, Valley Green, McGuards Dam, and Kitchens Lane, a large number of those were over the limits that were the guidelines from the EPA. And the second slide shows that anything in red indicates that there was an increase from a dry to a rain sample. So if rain had occurred within 24 hours, anywhere listed in red is the concentration, the median concentration. So you can see that many of the locations had increases in contamination after rain events. Specifically at the Devil's Pool and Rope Swing, which are popular recreation sites. And these were all over the suggested limits for recreation from the US EPA. And so what are the takeaways from our preliminary results? We found that 13 out of 20 rain samples had greater median FiO concentrations than their dry counterparts. And specifically, Devil's Pool and Rope Swing had the greatest increases in FiO markers after rainfall events. So it seems that recreation within 24 hours of rainfall could be riskier than during dry conditions. So special care should be made sure to avoid swimming at those times. And then Shannon is going to present part 2, which is translating this data into risk assessment using the QMRA approach.
Okay. So like Shane said, I'm going to discuss about what the next steps for this analysis will be. And I'm going to do that by actually kind of backtracking on some analyses we've done in the past, since we are still waiting on the lab data. So once we receive data back, we're going to get data on the concentrations of the different pathogens and sewage markers in samples. And we're going to be able to use these to calculate more precise risk estimates than what we'd be able to get relying on the fecal indicator organism. So I'm going to demonstrate how we did this for previous samples at CSO impacted sites instead of at the Wissahickon, and talk about next steps for the Wissahickon project. So quantitative microbial risk assessment, QMRA, that's the method we use to get our risk estimates. So it's a modeling approach to estimate the risk of illness or infection after exposure to pathogens in the environment. So QMRA has 5 steps. The first 4 are hazard identification, exposure assessment, dose response, and risk characterization. Those are the steps I'm going to go over today. Then the risk management step is really just using that information to try to reduce risk later on. And you can use that step to change the different parameters of the QMRA and see how different types of risk strategies will actually reduce the risk that we're able to calculate. So this study was called Assessing Risk of Waterborne Disease Acquired Through Recreational Exposure to CSO, or Combined Sewer Overflow Impacted Waters in Philadelphia. And this is a map of the 3 study sites we used for this project, which was Cobbs Creek and Tacony Creek, which we also sampled in summer 2020, and then the Delaware River, which we didn't sample in summer 2020, but we sampled 2017 to 19. So I'm going to go through the different steps of the QMRA and show how it applies to this project. I'm kind of moving through things quickly for lack of time, but I'd be happy to answer questions afterwards. So the first step, hazard identification. So hazard identification step is used to say, what types of pathogens would we expect to result in risk of illness if you're exposed to them at these sites? And what concentrations do these pathogens exist at in the water? So we collected samples during both CSO impacted, which was less than 24 hours after a CSO, and non-impacted more than 24 hours after a CSO conditions at all 3 sites. And we used the samples to quantify concentrations of HF183. Because the risk of AGI, which is what we're interested in, acute gastrointestinal illness, may be from multiple different types of pathogens. And we did not have pathogen data for this risk assessment. We used HF183 concentrations to estimate concentrations of reference pathogens. So we chose 5 pathogens. some of which we found in previous data collection, and some of which are just very common causes of recreationally acquired waterborne disease to conduct the QMRA. So to analyze samples for HF183, we collected GRAB samples and analyzed them using qPCR at our lab at Temple University. And as Shane said, HF183 is a marker for human Bacteroides, which is a harmless gut bacteria that exists in really high concentrations in sewage. So it's a marker that indicates is very sensitive to human fecal contamination. So if we're finding high concentrations of HF183, we can be pretty confident that we are seeing human fecal contamination, which is a key source of human pathogens at the sites. So to estimate the concentrations of the 5 reference pathogens in our samples, we use this formula. So we plugged in the concentrations of HF183 in samples we measured during CSO impacted and non-impacted conditions, divided that by the concentrations of HF183 in sewage from the literature, and multiplied that by the concentration of each reference pathogen from the literature to get our estimated concentrations of each pathogen in samples. So this equation is kind of an oversimplification of what's actually going on because all of the inputs are probability distributions, not point estimates. So we're able to use that to account for variability in each of these inputs. The next step in the QMRA is the exposure assessment step. So for the exposure assessment step, we collected observational data at the same time that we collected the samples. So we know that the recreation was occurring at the same time where we measured that concentration of HF183. So exposure assessment involves collecting data that tells us the volume of water somebody might ingest during one exposure event. So to inform that equation, we collected data on the types of activities that are going on at each site, the number of individuals that are observed engaging in each type of activity, and the duration of activities. Using this data, we're able to calculate the volume of water ingested, which is the ingestion rate per activity that comes from the literature for each activity we observed, times the duration of activities we observed. And using the volume of water ingested, we can get the dosage or the concentration, the amount of pathogens one person ingests by multiplying the volume of water ingested by the concentrations of pathogens from the hazard identification step. Again, these are all probability distributions, not point estimates, so it's a little bit more complicated than just these formulas alone, but they give a good idea of how the model was put together. Next is dose response. So the dose response curves came from the literature. We selected the best curve for each pathogen based on study methods used in previous QMRAs, et cetera. For norovirus, which contributes a lot to the risk, we ended up using 2 dose response curves, and that's a little bit in the weeds for the time I have left, but the dose response curves for norovirus really heavily impacts the risk. So I want to point that out before I present the results of the risk characterization. So the risk characterization is the step where we pull all our inputs together. We use Monte Carlo analysis to sample from each probability distribution 10,000 times, and we're able to get a mean estimate and confidence intervals around that mean for each exposure event, the risk of illness for each type of exposure. So briefly, I'll just show some of that, what those results look like. So these are the results at Cobb Creek, where we observed a wading and fishing exposures there. So as you can see, these are the risk from at least one of the 5 reference pathogens we included. The risk using norovirus model 1, which is what's shown on the left side of the plot, is always higher than the risk using norovirus model 2, and that's because these are kind of 2 ends of the norovirus dose response spectrum. So we'd expect that difference there, but probably the truth lies somewhere in between is what they say. So the black line also indicates the US EPA threshold of 30 illnesses per 1,000 exposed, which has been used as just a general risk threshold above which we'd consider that an unacceptable risk. So we can see that using norovirus model 1, all exposures resulted in mean risk higher than that threshold, and using norovirus model 2, the CSO impacted boxes overlap that threshold. These are the results at Tacony Creek site. At Tacony Creek, we observed swimming, which results in a higher risk because of more ingestion, but similar patterns are observed. And then at the Delaware River site, we saw really similar results to Cobbs Creek. Delaware River is where we saw the lowest concentrations of HF183 across the sites, but still we would expect risk of illness during CSO impacting conditions above that threshold, especially during a fishing event, because fishing lasts a long time is why that difference is there. So what can our results tell us? The results tell us that the highest risk is due to a swimming exposure at the Tacony Creek site during CSO impacted conditions. They can tell us that the risk of illness is higher within 24 hours after a CSO. And they can tell us that a lot of scenarios resulted in a mean risk of illness for at least one of the reference pathogens above the acceptable risk threshold. So the next steps is to do the same process at the Wissahickon sites. At the Wissahickon sites, we'll get pathogen data in addition to sewage markers. So hopefully we'll be able to take the QMRA a step further there. And then we can use the Wissahickon data to compare to the sites where we've done our past work that are impacted by CSOs, which will give us more information about how much CSOs contribute to health risks due to recreation at the sites. So me and Shane would like to thank the undergraduate merit scholars and student workers who helped in sample collection and collecting observational data, and then the Philadelphia Water Department and William Penn Foundation for providing funding and assistance in different steps of the project. So we are ready to answer questions.
Excellent. Thank you, Shane and Shannon. That was really great. We do have a minute or two. If anyone has questions for our two presenters here from Temple, Shane and Shannon, or frankly, any questions from the other session, the other presentations in this session, we could take a few minutes to put them in the chat, or we probably have some time to ask them in person if you'd like. So we have a question from Charles. How did your pathogen-based QMRA compare to indicator-based risk estimates?
Thanks for the question. So I know that the previous QMRA that was done using enterococcus found pretty similar results to ours, except we did find a higher risk at the Delaware River site compared to that done using the enterococcus-based QMRA. So I think they were pretty similar overall, overlapping.
All right. Well, here we have something from Nathan. Such a big difference in those virus models. Can you say more about why that is?
Yeah, I mean, I assume that, Nathan, that you're talking about the difference between the two neurovirus dose response curves. So the models using the first dose response curve versus the second. So those two curves use really different assumptions around what went on during the norovirus studies that collected the data for those curves. And norovirus contributes a lot to these risk estimates because it's really prevalent in human sewage and it's really, really infectious. And yeah, the norovirus dose response models are a mess, as Dr. Haas said. So I think that for now, using different curves and talking about how the limitations of each one and the assumptions made is probably one of the best ways we can go about modeling that. And then the question on the epi reporting data, we don't really have good enough epi data to be able to tell us which one is more accurate. So I did go through the reportable disease data, which was all that I had available. I tried to use some multipliers accounting for underreporting and underdiagnosis. And all I could say really is that our results for both curves were less. And Heather points out that, yeah, there's no epi data for norovirus. The reportable diseases I used were crypto, giardia, and E. coli.
Excellent. I think, oh, we do have another question here. This is great. From Melissa. What amount or duration of rain was needed in order for it to be a, quote, rain event where you would go out and sample?
So we used a really small amount. I think it was 0.01 inches, which was about 2.5 centimeters. I'm going off memory, sorry. I'm pretty sure that's what it was, but yeah, it's kind of a small amount, but we looked at some of the CSO data that the water department provided us, and we did see that CSOs were recorded when we saw small amounts of rainfall. So we ended up going with that small amount.
Okay, and Rafi asks, do you have any plans to share your results with PADEP, Friends of the Wissahickon, and other agencies or groups associated with monitoring of the Wissahickon? Seems like there would be real value in sharing these results with those partaking in primary contact recreation. And it looks like Heather answered.
Yes.
Workshop. Wonderful.
Yeah, so the Friends of the Wissahickon helped us pick out our sampling sites, which was super helpful, and they've been really involved in the process. And we're definitely planning on presenting this once we have results and we are able to do the modeling. We're hoping to present this to a wider audience.
Great. And since we have a couple minutes, we have, if anyone has questions for the prior presenters, we can take some of those also. Or again, you can contact us separately and we'll forward your questions along to them. And seeing nothing in the chat, I think then maybe we can wrap up a few minutes early, which is always a good thing. On behalf of the Academy, I would really like to thank everyone today for attending the 4th annual Delaware Watershed Research Conference. I'd like to especially thank all of our presenters and the William Penn Foundation, who is our sponsor for this and for so much of the work that was presented here today. And a special thank you to the folks from the Bees seminar for joining us. We really hope to welcome you back to the Academy for next year's conference in person. We miss seeing all of you and having these great conversations one-on-one. But until then, you know how to reach us, and we hope everyone enjoyed all the presentations today and learned a lot. Some really exciting stuff going on. So thank you, everyone. That's a wrap for the day. All right, everyone stay safe, and we'll see you soon.
Thanks, Catherine. If you haven't registered yet for the conference, register. That's how I'll be distributing the video, the recording of the conference. So yeah, thanks to Catherine and all of our moderators because it ran very smoothly today. So thanks, guys.